Hello, I would like to extend my warmest welcome to everyone and say thank you. We are very happy you could join us today for the Microbial Insights webinar. Microbial Insights is delighted to have Microbigals today, Tess Dayet, John Mitchell, and Julie Gruba for a very special edition of the Microbial Insights Halloween webinar. Three monstrous microbes that mimic our most ghastly and glorified Halloween horrors is going to be today's presentation topic, while Microbial Insights celebrates its 30th anniversary. Dr. Tess Dayet, PhD, is a passionate bioinformatician with nearly a decade of experience in microbiology and microbial genetics. She is a strong advocate for science communication through playful writing and storytelling, which is showcased on her website, microbigals.com, and podcast, The Microbe Moment. She aspires to help discover and leverage the powers of our microbial friends for the benefit of the world. John Mitchell is a microbiome researcher at a pharmaceutical company. He received his master's in microbiology from the University of California, Riverside. In his free time, he enjoys home brewing meat. As the co-founder of the Microbigals, he produces, edits, and is the primary interviewer of the podcast, The Microbe Moment. And Julie Gruba is a senior application analyst and Microphil. She has a biology degree from the University of New Hampshire, a black belt in karate, and she's climbed all 48 4,000 footers of New Hampshire. She loves all microbial food and has swapped out coffee for her homemade chaga tea recipe. Recently, she even started to make her own kombucha. Julie loves to write and is the primary content creator at Microbigals. We are very excited to have them. I also have to mention that there will be a recording of this webinar available in the resource section of the Microbial Insight website within the next week. All registered participants will also receive an email containing the link of the recording. Moreover, you can find the webinar recording link on the MI social media accounts, including the MI Twitter, MI Facebook, and MI LinkedIn. So please be sure to check them as well. We will proceed with a brief question and answer session after the presentation. We encourage you to type your questions throughout the presentation, and our speakers will answer as many as possible at the end. So please let me turn it over to our speakers without further delay. Tess, the floor is yours.
Great, thank you so much. We are super excited to be here. Just gonna stop sharing my webcam. Great, all right. Now, so we're going to start with a little bit of an introduction. This is going to be sort of a fun webinar, I hope, where everyone's going to learn a little bit something about microbes in Halloween, as well as some fun facts that you can take to some of your Halloween parties over the course of the weekend as we get into Halloween weekend. So we're going to start a little bit of an introduction. I'm going to tell you a little bit about who the Microbigals are. So I am Tess. I have a PhD in bioinformatics, and I'm joined with John Mitchell, who is a microbiome researcher, and then Julie Gruba, who works in medical records. And then finally, we have Celia Shi, who's not here with us today, but she is one of our main artists on the site, and she does a lot of these little fun microbe cartoons that you'll see throughout this presentation. So I just wanted to give her a shout out as well. So before we get started, we just want to say thank you to all of our followers. Thank you to Zymo Research, who has sponsored our podcast over the past 2 years. We really have enjoyed building this community and sharing some fun facts about microbes to the world. So beyond Halloween, Microbigals is science stories and news for the microbially enthused. We are a science blog and podcast aiming to show everyone and everything has a unique microbe moment. We have blogs and podcasts that touch against everything as microbes touch everything, from history to interviews to recipes. We call them Fungal Foodie Fridays. To nature, we do a lot of hiking. We're all very much outdoor enthusiasts, and sustainability as something that is near and dear to my heart as well. So on the left here, and this is just another little tidbit of showing our little bit of our nerd flag, we have the back batch of microbes. So we do a lot with sci-fi, particularly with Star Wars, and kind of connecting the microbial world into the science fiction world as well. And in fact, our next little segment at Microbigals, we're diving into astromicrobiology, and we're going to do a podcast connecting some of the big sci-fi moments that uses microbiology, either to villainize or to be heroes in the storyline. So that one's going to be a lot of fun, I think. This is just a little collage of some other things that we can do. There are some famous scientists that you'll see here, and maybe some scientists or microbiologists that you're unfamiliar with. We do really try to showcase Black microbiologists, female microbiologists, microbiologists that are more or less forgotten in history. But we do think they've done some awesome things, and they need to have a little spotlight. So we have scientist spotlights as well. Okay, and then this is my final little thing on Microbigals before we get into what we all came here today on our Monstrous Microbes. So this is the Society of Symbionts. This is our little school in the microbial world, training the next generation of microbes to love, care, and share with the giants, which is ourselves. So they teach classes like criminology and pathogenesis, so that's taught by Becky Lafage, to extremophile survival, which is taught by Coach Timmy Tardigrade, into things like environmental sciences, which is taught by Miss Mycorrhizae. So each character, each teacher is based on a real microbe and teaches a class that helps to even educate ourselves on what the microbial world does for humanity. Okay, and then finally, we're going to get into our monstrous microbes. And I think each of these three tales, we have three separate tales, three different microbes that have three different unique microbe moments for Halloween, but they all have one underlying lesson. And that is the only thing to fear is the knowledge that we do not know. And a lot of times when we're dealing with the microbial world, we can't see them, we don't know what's going on. And in particularly our historical stories we're going to tell you today, they had no clue what microbes were, and they brought a lot of fear, a lot of imagination into their lives to the point where things got a little crazy. So today we're talking about three stories: vampires, zombies, and witches, and their micro moments that are associated with them. Actually, many of these have multiple micro moments, but we'll be highlighting a single story or maybe a couple stories within each of these on how microbes relate to some of these Halloween characters. So without further ado, I'll hand it over to my co-host, Jonathan Mitchell, to talk about the Great New England Vampire Scare.
Hello, everyone. So, like Tess said, I'm talking about vampires today, and this focuses specifically in the New England area. Now, there was this outbreak, you could call it an outbreak of vampires between the 18th and the 19th century, where cases occurred where families ended up desecrating the bodies of their deceased loved ones, thought to be vampires feasting on their surviving members. Some bodies were just flipped over, others had their hearts cut out and burned, and even more, some were cremated or decapitated. And all this was done to stop the predatory actions of the vampire and to save the remaining family members, people who were wasting away and looked like they were having their life drained from them. How these, it was thought that the supernatural was bringing back the dead to feast on the living, but actually it was tuberculosis that was leading people to the graves. In fact, 12 of the biggest vampire-related exhumations in New England were due to tuberculosis, and even Henry David Thoreau had mentioned these vampire cases in his journals. So I really want to highlight three of the more famous cases that occurred in New England. First, we're going to talk about the Brown family. This occurred in Exeter, Rhode Island in 1892. Several family members had died of the disease, leaving the father George and his afflicted son Edwin. But Edwin had developed tuberculosis and had gone to treatment in Colorado, but his condition worsened. And George was encouraged by his community to dig up his family and find the vampire, which would be one that had uncoagulated blood in their heart. Now, one of his daughters, Mercy, was 19 and had died 2 months prior and had not been buried due to the winter. And upon inspection, blood did end up seeping out of her heart. what that ended up being was decomposed clotted blood, but for them it confirmed that she really was the vampire. So what ended up happening is her heart and liver were removed and were cremated, and in a last-ditch effort, put in a tonic and fed to Edwin. But unfortunately, this didn't work, and he passed 4 months later. The second family is the Ray family, and this occurred in the Jewett City, Connecticut in 1854. Again, their son was afflicted. This was Henry. And the family had members dying of the disease over a 9-year period. What they ended up doing is exhuming 2 of his other brothers. And what they ended up doing was burning the bodies or part of the body. And Henry ended up recovering. And because of his recovery in that area, burning of so-called vampires became the standard in the area. Last but not least, we have Griswold, Connecticut. And this really happened, this started in 1990, where these graves were long forgotten, but kids had found them playing in a quarry. And they originally thought it was a burial ground due to a serial killer in the area, but when officials got there, they found it was an old colonial family. A crypt at the gravesite revealed a skeleton that had been decapitated, with the head placed along with the thigh bones on the ribs and the vertebrae. And the vertebrae had been broken, likely due to someone trying to remove the heart. However, this body they found had been in the ground for at least 5 years. There's not really much left, so they relied on decapitation to get rid of the vampire. So I want to talk a little bit about tuberculosis, or Mycobacterium tuberculosis. It's a rod-shaped, non-motile, acid-fast bacillus. It grows very slowly, 15 to 20 hours generation time, and can cause the disease tuberculosis, typically infecting the lungs. And this is classified either as latent TB infection or active TB disease. So latent TB is a state of the body, particularly where macrophages in the lungs prevent the growth and spread of the bacteria by forming a granuloma. And it can become active when a person's immune system becomes compromised, such as someone that's infected with HIV. And then this develops into active tuberculosis, with the symptoms including persistent cough with blood or sputum, weakness, fatigue, weight loss, night sweats, and fever. And treatment of the active TB depends on the bacteria susceptibility, with susceptible TB including cocktail multiple antibiotics for 6 to 9 months. And drug-resistant forms are much more complicated, and if treatable, can take up to 30 months of antibiotic treatment. I will say that there can be antibiotic treatment for latent TB, but it's much shorter. So I'd like to get a little bit into the history of tuberculosis in itself before we wrap back into vampires. It's believed that this bacteria has been associated with humans for 15,000 to 20,000 years. And it can be found in Egyptian mummies and that have been buried since 2400 BCE. And the names can be of the disease can be found in text from India over 300 years old. And it has gone by many names, such as Phthisis in Greece, Thebes in Rome, the White Plague in the 1700s, 1800s, consumption, and my favorite from the novel, The Life and Death of Mr. Badman, where the disease is called the captain of all these men's of death. And treatments were developed over the centuries, with the most well-known being sanatoriums, where people are placed in areas that tended to be dry and were thought to have clean air, which was supposed to help the disease. And of course, March 24th, 1882, the existence of bacteria was announced by Dr. Robert Koch, with the first TB vaccine, the BCG vaccine, being developed in 1921, and then the first antibiotic treatment with streptomycin in 1949. So I will say that I have a little addendum here. It says that vampires originated in Europe and were not exclusive to tuberculosis. This is true for tuberculosis, but if you look into many cultures around the world, vampires seem to pop up almost everywhere. And some of them are what we think, like when we think of Dracula, and some of them are almost kind of like this mindless zombie going after other family members. And in Europe, a lot of times vampires were associated with pestilence and disease. And disease would suck the life out of the afflicted and bring the so-called vampire back to life, or it would allow them to rise out of the grave once they had fed enough. In fact, there was a body that was found in the 16th century plague pit. It had a brick in their mouth due to prevent them from feeding on the living. And this was actually what they call the vampire Venice. And this person was discovered in 2009 in Italy. And in New England, of course, this was associated with consumption, but in America, this wasn't the only type of vampire that we saw. Early days of New Orleans, they needed, well, there was a large male compared to female population. They brought over many women to try to increase the colony's population size, but these women ended up suffering from scurvy along the travel to America. And when they came off the boat, they had sunken eyes, their skin was pale, and it seemed as if they were bleeding from their mouths. And some ended up believing that they were vampires, and there are tales or stories of them being quote unquote locked up in a covenant in the area. But bringing it back to New England, so the Brown case occurred after Robert Koch's discovery. And I believe this is due to news. I mean, the area, New England at this time was very rural and news traveled very slowly. So sometimes it could take over a decade for new treatments, news to reach certain areas. Not to mention, your family members are dying. So what are you going to do? You're going to try everything possible. And this seemed to be the last ditch effort. And so you're going to try to save the rest of your family. But eventually this practice stopped, and today there have been at least 80 quote unquote vampire bodies discovered in New England, with the likelihood of more out there. But that is it for New England vampires, and I'd like to turn over to Julie to talk about zombies.
Good morning, everyone, and happy almost Halloween. I have a very scary story about real life zombies that's probably happening right now somewhere in the world. Our story opens with Annie the carpenter ant, and she's just walking along the tropical rainforest floor somewhere in Thailand, doing what she always does. She's gathering a leaf to bring back to her colony, just doing her part to ensure the survival of her ant family. On this day, though, her whole world is going to change. She couldn't see the microscopic spores that she innocently walked through, but they attached themselves to her legs. She continued on with her chores, oblivious to the horror that was occurring. And as she was working, those spores were infiltrating through her tough exoskeleton and were beginning to slowly eat away at her organs and seize control of Annie's muscles and interfering with her nervous system. A couple of days later, Annie descends from her tree nest for the very last time. She wants to do her job, but she feels like she doesn't really want it. She's not going the direction she wants to go in. Something is kind of guiding her somewhere, and she's not sure what's going on. And she's not in control anymore, and she doesn't know where she's going, but it's about to get worse. She finds herself climbing up to an underside of a leaf, and it's a very specific spot. It's on the underside. It's 25 centimeters high. What is she doing here? She doesn't know. But suddenly she feels an undescribable urge to clamp down on the vein of an underside of this leaf. And she can't move and she doesn't know what's going on, but she clamps down and all of a sudden her world turns black. And this is probably a good thing because the fungus that has infiltrated her is going to continue to eat Annie's organs and use her lifeless body as protection from the elements as it continues to grow. To add insult to injury, this now grows a antenna-like appendage out the side of her head, and that is the fungus has grown enough, and it now is ready to replicate itself, and it releases spores out of that antenna, and she is now, providing nutrients for the fungus. The fungus has grown enough, it wants to spread around, and so it pops out of the back of her head and ends up releasing more spores, which float down to the forest floor, and then another unfortunate ant is now infected with the same thing that our poor Annie was. And this is a fungus called Ophiocordyceps unilateralis. And even with this kind of gross way that it makes a poor ant into its own little zombie, humans actually consume cordyceps, probably not this exact kind, but it is out there, and I guess that's a little bit of fair play. The cordyceps make victims out of insects, and then some humans also will consume certain types of cordyceps. So what is cordyceps? This Ophiocordyceps unilateralis. There are actually a lot of different types of cordyceps. And this particular one was discovered by naturalist Alfred Russel Wallace in 1859, and it's generally found in tropical forests. But is the fungus actually evil? And this goes into what Elise was saying earlier. We make up stories to explain things sometimes, and when it seems like sort of an evil thing, we kind of give it monstrous characteristics. But the fungus isn't evil. It does have very specific requirements about where it wants to land, and so there's been studies out in the forest finding these, and they it's amazingly, they like 98% of the times when they find these ants, and you can see that picture there of what it may look like, they are always found kind of in the same place, 25 centimeters above the ground. They have a death grip with their mandibles into the vein of a leaf on the underside. So the cordyceps doesn't have any way to get itself there. And so this is a way that it has developed, which is kind of creepy and maybe not so nice to the ant, but it is pretty fascinating that they are able to do this. And it's been doing it for a long time. They have found ants kind of in this condition, maybe with a different type of fungus, but they have found this in pieces of amber that are 50 million years old. So this has been going around for quite a long time, and it's pretty amazing to think of the evolution of how this happened. So what happens here, and you know, it's not like you go out into a tropical rainforest and you find, you know, millions of ants clamped down on the bottom, but it is common enough that they're able to find it and study it. So the cordyceps is going to kind of start consuming the ant, and we'll have a couple of pictures coming up here of other cordyceps that take over other types of insects. So they're different species of the cordyceps, and they have kind of evolved to sort of take over and zombify specific insects. But interestingly enough, it does consume, you know, the organs, but it does not consume the brain. But what it does with the brain is even kind of more fascinating, and I think that they're doing a lot of studies on this because it's quite fascinating and could have some interesting applications. But they, through some bioactive, you know, chemicals and compounds, it interferes with the ant's nervous system and basically allows it to kind of control it to get it to this very specific environment that it needs to grow. So it compels it to go there, and there's humidity, there's the height for some reason, the location of the underside of the leaf and into the vein. And when it gets there, it ensures that it has a place to stay and that it's safe, and that's why it has the ant go to that certain spot, and it makes they have found that it makes the mandibles, which are the you know, basically the front teeth of the ant, clamp down, and it's an unnatural movement. What they have found, like the way that it clamps down in this particular case when it's taken over by this fungus, is sort of an unnatural way to clamp down, and the ant does actually die at this point, where it doesn't need to go anywhere, so it's no longer needed alive. So it dies, and the ant will continue, or the fungus, sorry, will continue to consume the ant, use that nutrients in this kind of protected environment, and it continues to grow. So it is going to, once it gets to a certain point of growth, it will extend itself out of the ant's exoskeleton. And we have a couple of pictures there where you can see that it grows like this antenna-like appendage, and you can think of it sort of like an apple on an apple tree. You know, that is the way that it's going to spread its seeds, and in this case, it's spreading spores. And if you are not aware, when you see mushrooms out in the woods that have grown up out of the ground, or maybe have grown out of the tree, that's only a very small part of that fungus. It's actually its hyphae, which are, you know, generally some very thread-like, you know, kind of mapping of these threads that kind of spread out, and when conditions are right, it sprouts a fruit. In the case of cordyceps, it's that antenna. In the case of mushrooms that are out in the woods, you know, that have popped up out of the ground, that is basically a fruit, and those fruits are going to release spores, and that is how the fungus spreads to new areas. So as I mentioned, ants aren't the only ones who are become zombies at the behest of fungus. Moths, wasps, spiders, caterpillars, beetles, flies, cicadas can also fall prey. If you see that little beetle right there, if you look closely, you'll see that it is hollowed out. There are no organs in there, but the thing is still moving around, which is pretty horrific if you think about it, that a fungus could actually get in there and control the muscles to keep a bug moving around. And you can see in our examples here, you know, these all have, kind of sprouted, and so these are, spreading more of that spore around. And we'd like to maybe, monsterize these fungus, but they do play a vital part in the ecosystem. Fungus are in charge basically of, cleaning up forests. They eat the wood, they eat all kinds of other things out in the environment, and help to keep things cleaned up. Because if you think about it, if we didn't have fungus that were out there doing the work that they do, the forests would be piled high. We wouldn't even be able to walk in them. So they are out there. When you see them on trees, and you see them, they're actually eating those, consuming those, and then turning around and making more of itself so it can continue to do that. One of the other little tidbit we have here about the cicadas, the way that these fungus are able to adapt themselves. So a lot of the cicadas only come out every 17 years. And so there is a corresponding fungus that does the same thing. It can hibernate basically, and for that, because they are specific to cicadas. So they can lie dormant in the soil, wait for the cicadas to come out, and when they do come out, they infect them and start basically eating away at the body. And you can kind of see those. We don't have them here in New England, so I've actually never seen one. But you can see in this picture here, it looks like part of the body is gone. It kind of starts eating from the tail end, and you can see that it has it's like a white paperish, that is the fungus growing. And interestingly, it wants to keep spreading around. So the way that this particular cordyceps does that is that it makes a male cicada make the sound of a female cicada to attract other males. And so when that male comes around, it goes to mate with the infected cicada and ends up getting infected itself, and then the cycle continues. So pretty interesting and kind of scary to think about, that there's fungi out there that could actually control insects' brains to do its dastardly deeds. And with that, I will hand it over to Elise to talk to us about witches.
All right, guys, I hope you're still with us. We have our third and final tale to talk about. And this one is going to be sort of a combination of the last two that we did. We're going to talk about New England. We're going to talk about history. We're going to talk about fungus. Particularly, we're talking about the infamous case of the Salem witch trials. So, but first, before we get into it, let me talk a little bit more about microbial history and where we are in time. So we have Robert Hooke and Antony van Leeuwenhoek, who were both around in the 17th century, late 17th century, and were the first to sort of describe microbes. So Robert Hooke came up with his first scientific bestseller, Micrographia, which he described for the first time the macro world, the micro world, or the unseen world. Not long after, we have Antony van Leeuwenhoek, who started to publish his work, in which he was the first person to actually draw a bacteria in scientific literature. He developed over 500 unique lenses that he put in his microscopes in order to see things like yeast and bacteria that he found around, from everything from teeth scum to soil. And then finally, I just want to put this into another microbe moment of New England colonials, and that is the story of Onesimus. So Onesimus and smallpox outbreak of 1721 happened about 30 years after the Salem witch trials. But what's interesting here is a lot of the players in the smallpox outbreak of 1721, such as Cotton Mather's, was also a part of the Salem witch trials. So we do see New England going through a number of these trials in their early days, trying to combat microbes in a real devastating way. So in this story, I'll tell it kind of quickly. Onesimus was a black slave in New England, And while he was in Africa, he had this practice done to him called virilation, which is the practice of taking the pus of an infected individual and inoculating it into someone who doesn't have the disease. And the idea is if you just have a small amount, your immune system will then adapt and create antibodies and then be able to fend off the disease that you see. So this is sort of our precursor to vaccines. It's definitely not as safe as vaccines, but it was something that was used in sort of the early days before we developed vaccines and sort of the safety practices that we have today. So in this 1721 outbreak, 844 people actually died of smallpox. 242 were inoculated in this process of virilation by a doctor, and only 6 of them died of smallpox disease. But we're not here to talk about smallpox. We're here to talk about the Salem witch trials. So the Salem witch trials really start in January of 1692, where Elizabeth Parris and Abigail Williams started to have these really weird sensations. They would bark like dogs, convulse, vomit, spoke in strange languages, have feelings and sensations of things crawling all over their skin. It terrified a lot of terrified their parents. Elizabeth's parents was actually one of the reverend of the Salem village at the time. And he grew very concerned. He brought his child to the doctor and asked what was wrong with her, what could be done. And the doctor really didn't know. I mean, again, this is the time before we really understood what microbes were. This is a time where germ theory was not really well recognized. This is a time where they didn't really have any medicine. A lot of times, if you had anxiety, if you had some of these symptoms, they might send you home with lavender milk because lavender has this calming sensation, and that is something that we still sort of use today because the smell aromatherapy is so powerful with lavender. But this did not work for our girls, Elizabeth Parris and Abigail Williams. They continued to have these weird sensations. And so a neighbor decided maybe what they needed to do was a witch cake to determine whether or not these girls were actually bewitched. Now, a witch cake is made of rye mixed with urine from the afflicted girls. And the idea is that you bake this witch cake and then you feed it to the dog. And then because the dogs were associated with the devil. And so if the dog ended up having the similar symptoms as the girls, they would declare that they were bewitched. Now, this is some, folk magic, folk medicine for sure. And it's there are different records on whether or not they actually recorded what happened to the dog. Regardless, after they fed this witch cake to the dog, things got really crazy up in Salem. And everyone believed that they were bewitched. And this is when the finger started pointing. And in the end, this actually occurred over the point from January 1692 all the way to about September, October 1692. So we're talking about a 9-month period, right? In that time, they hung 19 people. They crushed one man to death. This would be the infamous Giles Corey, who would always say more weight until eventually he couldn't say anything anymore because he died. There was also 5 people that died in jail. And one of these people that died in jail actually died in jail many months after the whole Salem witch trial had ended. So in this time, you had to actually pay a renting fee on your shackles to be in jail. And when you were in these shackles, if you couldn't afford the rentals of your shackles, they would just keep you in jail until you could afford it. So one very poor woman could not afford her rental fee for her shackles and ended up dying in jail because of the terrible conditions that these jails were. We're talking dirt floors. We're not talking very good food, cramped quarters. It was not a very nice place to be at all. The youngest person to go in jail was actually a 4-year-old Dorothy Good, whose mom would also be accused of witchcraft. While Dorothy would escape the noose, her mother would not, and she was hung for witchcraft. And then finally, which I think is one of a very interesting tidbit in the Salem witch trial ordeal, is the fact that a lot of the accusers were girls under the age of 20. And we're talking Puritan New England, where girls under the age of 20 barely had a voice for anything. They were not, their opinions did not matter. They were just instructed to do very specific things and otherwise to just sit down and shut up kind of thing. So it was very interesting that these girls had such a power that would lead to such a craze in the Salem area. So there are kind of three prevailing theories as to what caused this mass hysteria, and one of them is mass hysteria. But the first one is the idea of property gain, political gain. So a lot of the judges, a lot of the police that were involved in this ended up gaining significant amount of land, significant amount of income and political gain through this that they were able to leverage later on in their lives. In particular was George Corwin, who was a 25-year-old police sheriff. A lot of people have accounts that he actually enjoyed a lot of what he was doing, which was shackling people, repossessing their possessions, selling them off, keeping them, whatever he felt fit. And there was a big feud that went on that we won't talk about, but is very interesting and is a curse on all Boston or Massachusetts sheriffs from all time, or so they say. The second is mass hysteria, which I think is one of those things that is really hard to wrap our heads around, because it's hard to believe that mass hysteria can kind of cause the death of 25 people in such a short amount of time. But we see this again and again in our society. We can go back to the 1980s with the Satanic Panic and the way that they ostracized huge groups of people then. We can go back to the 1950s with McCarthyism, where people again were pointing their fingers at people over communism, and this is where we get the crucible. And then you can even look back during the pandemic and all the finger pointing that happened, especially on the early days of the pandemic. And in each one of these cases, this finger pointing comes from humans not understanding something. And when we don't understand something, we get very fearful and we need something to blame. We need something to latch onto. And so this is the idea of mass hysteria. And then our final theory brings us to the microbe moment of the Salem witch trial. And this is Claviceps purpurea, which is a fungus. So this theory was proposed by Linda Corporeal in 1976 when she published a paper arguing that the Salem witch trials could have been the result of ergotism. So Claviceps purpurea is an ascospore picked up by the wind. It lands on the ovary of a plant and starts infecting it. It then starts growing a hyphae. This hyphae turns into a sclerotia, forms this home, this ergot body, which is a mass of fungal mycelium. The seed itself will end up being 1 to 5 times bigger than natural host seed. It will turn purple or black, and each sclerotia can produce up to 1 million ascospores that can be infected to a new host. These spores can then be transported through wind or through insects or through other various dispersion methods to infect the next host. And when you're thinking, what is the range? We're talking grasses, which usually spread out over a field, or cereal species as well. The most economically important one is rye, and rye would definitely have been in the Puritan Society of Salem at the time. We even saw the witch cake was made out of rye meal. So if there was ergot, if ergotism was a proponent of it, it definitely seems like if you're making a witch cake out of rye and trying to figure out if your dog's going to have the same symptoms, he could also be infected by ergot. But there are a number of evidence, both for and against, whether or not ergot had anything to do or something to do with the Salem witch trials. So let's talk a little bit about the evidence for the Salem witch, or for ergot being part of the Salem witch trials. The first is they had very similar symptoms. They had violent fits, they had dull eye trances, hallucinations, a crawling of the skin, which is known as formication. These are some of the symptoms that you can get from ergot. Not all of them, but it is some of them. You also have the fact that the weather was definitely perfect to spread ergot. Most of the accused were in West Salem, where it was marshy, it was grassy. They had an unusually wet and warm summer of 1691. So this would have been the rye that they would harvest in the fall, would have been the rye that Abigail and Elizabeth would have been eating in January of 1692. So it definitely could have started some of these sensations that they had. And we also see that rye is an important source of food for early New Englanders. So in that sense, yes, ergot could have played a role in the Salem witch trials. However, when you really look at the Salem witch trials, there's also a number of evidence that points to ergot not being the primary source, or maybe not even a source, of the Salem witch trials craze. The first is that it would seem to turn off and on again, which I think is an interesting thing that people talk about, because ultimately people are only recording evidence inside the courtrooms, and it definitely seemed in the courtrooms when they were trying to figure out who was being accused, they would definitely point the fingers and have these sensations or see the specter of the people they were accusing at that moment, but maybe not when they were out in the streets or when other aspects of the court proceedings were going on. And finally, there was a very sudden and direct halt to the Salem witch trials, particularly when they started pointing fingers at the governor's wife. At that point, the governor said, okay, Salem, knock it off. You're all done. You need to stop this silly business of hanging people. And they did. And he saved his wife in doing so. Okay, so I want to end this little segment on Claviceps purpurea by talking a little bit more, some fun facts about Claviceps purpurea and other aspects that it influences our lives today and in the past. So the first is that Claviceps purpurea is or ergotism is a disease that has been a part of human society for as long as we've been as long as agriculture, really. And through that time, it has come up with a number of different names. We have super rye, we have the holy fire, we have St. Anthony's fire. And again, many people may not have known that this came from rye because while the seeds, you can obviously see they look different here when you're harvesting a whole field, you may not notice it at all. And then ergot, or Claviceps purpurea, has a number of alkaloids that we've seen in medicine as well as in psychedelic history. So in April 19th, 1943, we have what is known as Bicycle Day. It was founded by the chemist Albert Hofmann, who accidentally discovered and dosed himself with LSD, which stands for lysergic acid diethylamide. Lysergic acid is an alkaloid of Claviceps purpurea, while it does take a lot of massaging to get to the LSD form from Claviceps purpurea. However, this does open up a lot of psychedelic history in our world, and psychedelics have, and what's really interesting is when he went home, he didn't know that he dosed himself. He didn't know anything about psychedelics, really, and he asked his neighbor for a glass of milk. When his neighbor came over, he said he didn't see his neighbor. He only saw a hideous and horrifying witch, so which I think is kind of interesting that ties it back to our Salem witch trial story. And then finally, Claviceps purpurea has been known in a number of pharmaceutical recipes as well throughout history. It's been used, or the alkaloids that have been derived from it have been used to help migraines and cluster headaches. They've also been used for a number of pregnancy abortions, inducing childbirth, and even in aiding dementia. So I think there's a lot of other research out right now about psychedelics and about fungus and the alkaloids that are there and how we can use this and massage these sort of alkaloids into medicinal properties. So that is the end of our show. I want to thank Microbial Insights for having us. I want to thank everyone for sticking with us to the end. And if you really did like this sort of thing, we do have a number of other Halloween horrors that we highlight in blogs and podcasts. So the one that you see most prominently in the center of this slide is cavities, which is our true Halloween horror, particularly for our kids when they're going out and eating all this candy. But yeah, I hope everyone had a really fun time. And just as a final thing, we are doing a little bit of crowdsourcing right now, trying to come up with a podcast where we are highlighting everyone else's micro moment. So if you'd like to be featured in that, all you have to do is send us either a audio file or even just a text of what your micro moment is. Send it on to microbigals@gmail.com with the subject My Micro Moment, and we can feature you in our upcoming podcast. So thank you so much for listening, and we'd be happy to take any questions if you guys have, whether it's on podcasting or SciComm or anything else that we do at Microbigals or some of the Halloween horrors we talked about today.
Okay, thank you so much. That was an amazing presentation. I really enjoyed it. And we have so many positive comments about how fun and great the presentation was and how relevant it is to the Halloween topic. And so again, there are it's time for you. If you have any question, now it's time to ask it. So please go ahead and enter the question in your GoToWebinar panel under the question section, and we will get to them as many as possible. So the first question is, one of the attendees is asking that. So you mentioned for ergot the Salem trial, and then some reasons to believe it was ergot, or some of them like not to believe it's the source. So did they ever find out what was the reason causing these things?
No, yeah, they have no clue exactly what caused the Salem witch trials. I think most people believe it was a mixture of political gain and mass hysteria. Ergot may have played a role in the beginning. There's also a lot of theories out there on whether or not these girls were just sort of acting out, either because of child abuse or because of neglect. And they may have had ergot at the beginning, but then sort of like the attention they were getting and continue on. But it's one of those things that happened in the late 17th century, before records were what they were today, before medicine was what before we today. So I think this is going to go down as something we will never truly know what happened in Salem in the summer of 1692. But there are lots of different theories that you can latch onto.
Yeah,
Okay.
just to add to that, they did. I mean, they did have records, but actually, it was notorious for around this case that they got rid of them, or the judges destroyed like a lot of the writings of the proceedings.
Yeah, that's true. A lot of people, and there was the other thing is like your Puritans had a lot of shame. A lot of people had a lot of shame on this, and at the time, the best way to hide your shame was to pretend it didn't happen. So a lot of people, I think, washed a lot of records out, and a lot of people just didn't talk about it after it happened.
Okay, wonderful. And the next question is, somebody is asking that we talked about that fungus, cordyceps, if I'm right. So they were asking how it's being transmitted to the other species. Like you mentioned that they're spreading through like sprouts, and sometimes with cicadas it's like mating, but is it like airborne?
Yes, so the spores are microscopic, and they are, basically that fruiting body. In the case of the cordyceps and the ants, is that antenna that kind of comes out up through their skeleton, usually in the head area. It comes out, and these microscopic spores are released. And they are generally airborne, and in that case, they kind of float down to the floor of the forest, and they're picked up by another ant. And so similarly with the cicadas, it's you can kind of think it's not exactly a powder, but it's these microscopic spores that then are kind of on the legs and the body parts, and they kind of just get spread to each other via contact or airborne.
Thank you. So a relevant question I just received now is they're asking if this fungus can also infect humans.
No, these would not. They obviously they're microscopic, so we could come in contact with them, but we obviously would not. They aren't designed. They don't have the ability to infect humans the way that they infect insects. As a matter of fact, if a different type of insect walked through where where the ant walked through and it got infected, a different insect might walk through and might come in contact with it, but would not have the same result, because that cordyceps is very specific to whatever type of insect it has over time developed to interact with.
And cordyceps is something you can probably go and find in your local grocery store right now. A lot of fungal coffee, or a lot of vitamins right now are really looking into fungus as a way to boost your immune system or to boost your brain, to boost your thinking and memory. A lot of that is cordyceps, it's lion's mane, it's reishi. So these are things that you can probably find in your local grocery store, and they will definitely not turn you into a zombie.
Yeah, and there's thousands of species, they think, of cordyceps specifically, so that they are all a little bit different, and they may interact with different insects, or in our case, if people are ingesting them, they are different types of cordyceps.
Okay, awesome. So I guess we only have time for one more question. This is kind of like a general question from one of the audience, and they like mentioned that they will listen to other podcasts you made before, and then they have like lots of positive comments about it, and so they're asking if you have like any fun stories on top of your head about your challenges in making podcasts and how you're like coming up with the topics.
Ooh, that's a really excellent question. See how fast we can answer it. But like the topics as far as that goes, like it's just I think about microbes more than I'm awake, probably. I have dreams about them. And it's generally anywhere, anytime. We just went on a vacation to New Orleans. We go on vacations all the time, and I'm always like, oh, we should do a podcast about that. We should do a podcast about this. And so it's just something that I'm always thinking about, something I love to think about. And so, yeah, I just come up with all these. I have way more topics than we could ever record, which I think is really fun because I read a lot about how to make podcasts and blogs. And a lot of people say, yeah, if you run out of topics, like here are some things you can try. And I can't even imagine running out of topics.
Yeah, I'd say like in terms of challenges is learning how to edit and record. That was probably the biggest challenge. We've tried several different softwares, and we've picked ours. It's just like at the beginning, we're talking about hours upon hours upon hours, and just getting yourself a little bit better and getting faster. For me, that was one of the biggest challenges.
Okay, perfect. Thank you so much. We are reaching to the end of our time. I appreciate you all answering all these questions and sharing your knowledge, insight, and experience. We really enjoyed that. So in addition to our speakers' contact information, which is visible now on the screen, you can also contact us at info@microbe.com, and we will be happy to help you reach into Microbigals. I would like to thank you all for taking the time out of your day to participate in our webinar today, and I look forward to seeing you all again in the future. Thank you so much, and have a good day.
Bye, everybody. Thank you so much for coming.